So, good afternoon, ladies and gentlemen. I'm privileged to have everyone here who is attending my defense. I didn't expect to have this big room. I'm glad you have come to attend my PhD defense. The topic I'm presenting is host function and genetic loci associated with pediatric HIV disease progression in Botswana and Uganda. So, my supervisors are Professor Moses Gerald and Dr. David Katete. They're right here in case you don't know them. So I'll start with a small, brief experience before I start my presentation. If you have been around Uganda for the last, maybe coming to 25 years, in the early 90s, HIV/AIDS had just arrived here, and there were no treatment, what currently we are calling ARVs. So what happened then were people used to get infected, and the natural process of the disease will take its course on an individual who is infected, adults and children. So for mothers who are HIV infected, they would progress, they would go to the hospitals, deliver their children, and then these children have like two options. They either progress to AIDS very rapidly. So in my discussion, I refer to those babies who were born HIV infected, and there was no treatment, and they turned out to have the HIV disease very fast. So I'll call them rapid progressors. Then the other arm will be the long-term non-progressors. These were born to HIV-infected mothers, and they are also infected, but they stayed for more than 7 years without antiretroviral therapy, and they did not have AIDS. So those two will be a big component of my presentation. Thank you. So briefly, I want to talk about these two components. There's a group called the H3 Africa, which is also called the Human Heredity and Health in Africa, which is a consortium that is applying genomic technologies to understand different status in terms of health and disease among diverse populations in Africa. And this group also went on to have to allow people to apply these technologies in the different settings, studying different disease conditions. So still a project called CAFGEN, which is the Collaborative African Genomics Network, came on board to join the consortium, and its purpose was to apply genomic technologies to understand why children who are born HIV infected, some of them progress rapidly to AIDS, and others stay without developing AIDS for many years, at that time when there was no ART. So 4 years ago, I will take you back. 4 years ago, I saw an advert. They advertised to recruit PhD students to undergo a training that will enable them gain expertise to undertake this kind of studies. So I saw this call. I applied 4 years ago, and I was given an opportunity to take part of this. So when I read the entire project concept 4 years ago, I started writing this paper, putting different parts together. The concept of CAFGEN that was looking at utilizing genomic technologies to understand why some children develop AIDS very fast, while others don't, in absence of ART. So this paper was published last year, and it covers the entire CAFGEN project, looking at how it was conceived, looking at the research agenda, looking at how the institutions under CAFGEN were formed. It looks at the genomic infrastructures that were in place and those that have been implemented. It also looks at how the PhD students, of which I was part then, were recruited, trained in the US to gain this experience, and then how they came back and carried on the experience in analyzing the genomic data set that we are going to see. So this was, I'm happy to say, this was coined as a mark paper for CAFGEN, and I was the first author. I'm glad to be part of this. So it's the core paper when it comes to CAFGEN. It describes everything about CAFGEN. So let me introduce my topic at length. So HIV/AIDS remains a big problem on the continent, especially when it comes to the children. So this map is from the UNAIDS, and it was extracted from the period of 2008. This is data as current as 2008. Globally, there are 1.7 million children who are infected with HIV, and in that same year, we had 160,000 children who were infected, and 90% of those were in sub-Saharan Africa. So you can see how big the problem is. So during that same year, we had 300 children who were dying daily due to HIV/AIDS. So the problem is big, and it's here with us. So still trying to look at the estimates. Like I've told you, 4 years ago, I started my PhD. So what was my work relevant during that time? So here you can see from the data here, in 2014, for Botswana we had 111,000 babies or children who were infected with HIV. And in Uganda we had 69,000 who were infected with HIV. However, that was the time when I was conceiving to look at my research topic. So 4 years later, that is 2018, when I was looking at defending my PhD, was the condition still relevant? Still you can see the counts are not changing. We have 14,000 in Botswana last year who are HIV infected, and then for Uganda we had 100,000 children who are infected with HIV. So this work is relevant then and now. So it has not gone unnoticed. UNAIDS and WHO has come up with a very ambitious target, which is called 90-90-90. So what does it mean? It means by 2030 they want to end the epidemic of HIV/AIDS. But what have they done? They want by then, to have all the people, 90% of the people, knowing their HIV status. Just imagine we are 100 people, they want 90% to know their HIV status. Then, of those who test positive, they want 90%, at least, that's more than, they need the minimum 90% to be on treatment, antiretroviral treatment for HIV. Then out of those who are on antiretroviral, they want over 90% to have their viral load suppressed in that way. So that is the ambitious target by UNAIDS. And where does my work come in here? It comes in at this point. I would be much interested in identifying, after identifying individuals who are HIV infected, we can know who will progress rapidly and then who will not progress rapidly. So just imagine at one point you walk into a clinic and they tell you you have HIV, God forbid, and they tell you you can stay for the next 10 years without treatment. Yep, like we all know, our health care system is overstrained by delivering ARVs. There are many stockouts everywhere. So for individuals, when you try to talk about the coverage of ART, it is less than 80%, which is the target for WHO. So if we can be able to prioritize treatment among those who are in most need of that, would be glad to do that. So my work comes in right here, after individuals testing HIV positive, can I tease them out in 2 cohorts. So still looking at the HIV disease progression, there are 3 factors that come in to affect whether you progress rapidly or you take time. So one of them is the variation in the sequence of the virus itself. The virus has genetic variation, which is a factor of so many other contributing factors. But also the environment. Here the environment, what we need, things such as nutrition, lifestyle, and so on. So that will also come in to affect whether you progress rapidly or you take time. Then also host genetic variation. Your genetic makeup will also determine whether you progress rapidly or not. But it is also affected by race, gender, and age. So looking at these factors, they give us 2 broad phenotypes that we are interested in for this kind of work. I was interested in looking at the long-term non-progressors and then the rapid progressors. These are unique phenotypes that I've introduced in my introduction. So there continues to be a lot of inter-individual variability when it comes to HIV acquisition, disease progression, and death. There are individuals who are unique again, who fall off this. There are those who get repeatedly exposed to HIV, but they remain uninfected. So that's a unique group of individuals who many studies have identified particular host genetic factors to be responsible for this phenotype. Again, so many genomic studies have been done to try to identify what genomic factors affect whether people progress, delay the progression to AIDS. And these markers you are seeing here have been identified to be associated with delayed AIDS progression. And I've reviewed many of them in a paper that you can see in the next slide. So this paper will try to analyze and identify, ever since the introduction of high throughput sequencing, what has been done to ensure that they identify these markers. Unfortunately, in this paper, as we shall see in the next slide, we have very few studies that have been done to identify which genetic markers are associated with the delayed HIV progression. Yet these markers are very important for disease progression and also for advancing care and therapy for HIV-infected individuals. So this unique status I'm introducing here is called the long-term disease progression, not the long-term non-progressors. So in any population around the world, just imagine the way we are seated here, if God forbid we all got HIV infected, there will be a particular group of people, about 15%, who will stay without treatment. They will contain the disease for some good time. more than 7, 10 years, even 15. No treatment, but they contain the disease. So, if you have to look at the African continent, we have a region called Sub-Saharan Africa, which is a group of about 32 countries. So, in that region called Sub-Saharan Africa, we have 26 million HIV-infected individuals. And the ART coverage by that time was 53%, meaning we are not even anywhere close to a target, which is 80%. And 2 years ago, WHO launched a very ambitious plan called Test and Treat, meaning you just have to walk to the clinic, they test HIV positive, you return home with a treatment. And this treatment has its own side effects. And in Uganda, we have 1.2, as per last year, we had 1.3 individuals who are HIV infected. And if you have to draw these proportions, that means we have 195 LTNP individuals out of the 1.3 million infected individuals. But in Sub-Saharan Africa, if you have to extrapolate that same percentage, you will have 3.9 individuals who are LTNPs. So there is also again another small group of individuals. In the population, they are about 1.4%. Always they turn out to be a category called the elite controllers. These ones will control the HIV for many years. So if you add this count plus that means in Uganda we have about 200,000 LTNPs. That's a big population that you can harness to identify genetic factors, and then you are able to identify the markers in this big population and then advance care and treatment. So there's a study that was done by a group from Johns Hopkins in 2009, and they identified it was done in Rakai, one of the areas that has been highly infected by HIV. And they put the LTNP in that region was just one district, 9.1%. So many genomic studies have been done, like you have said, but 90% of those ones have been done in the Caucasian population. And they have identified particular genetic mutations that are associated with delayed progression. And when in cases where they have tried to replicate these markers in Africa, they have failed to replicate them. Yet we continue, like from the previous slide you have seen, we continue to have a big burden of individuals who are LTNP. They try to check these markers among this group and they can't be replicated, meaning we have a different signal that is responsible for this condition. Genomic studies that have been done, here I have this from a publication I've introduced in the 3 slides. A few genomic studies that have been done in diseases that are affecting the Africans so much. You can see how slim they are. When it comes to HIV, you have 14 genomic studies that have been done. And then you can see the cancers, and then you can see the diseases such as schizophrenia. The genomic studies that have been done are so many. So this is unfair. We have a disease that is infecting about 26 million people in Sub-Saharan Africa. And only 14 studies have been done to identify the underlying genomic causes. So my problem statement is you identify these markers will lead to therapeutic advances that can increase the lives of individuals who are infected. And the few polymorphisms you have seen that have been identified to be associated with delayed progression have been done in Caucasian populations who carry less than 10% of this disease. So that is the problem here we are looking at. So even those ones that have tried to identify those populations cannot be responsible for the same condition among Africans. So this forms a problem, and that's why we embarked on doing this work here. So the rationale is to identify these protective alleles in these big populations. In Uganda, we have over 200,000 individuals who are LTNP. We can harness this for identifying our markers of interest. So this study was set out to identify host genetic markers that are associated with pediatric HIV disease progression. And the advantage of using pediatric populations are many two. If you did this same study among the adults are confounded by so many factors, such as multiple sexual partners. So if someone got infected by strain A today, and you are going to do this study 2-3 years later, I can bet if you find the same strain at that point, because the lifestyles among adults are terrible. But in the children, you can use this as a check. They are less likely to have multiple HIV infection. There are other things that may confound genetic signals in adults, such as lifestyles, such as maybe smoking, use of hormonal contraceptions. All those ones will confound the signal. So then, secondly, children have so many years to live. When you develop a vaccine that will prevent people from progressing to AIDS, children will benefit more. An adult who is around 60 years, they may have maybe 10 years to live if you are to develop a vaccine for adults. But if you develop a vaccine for children, a child of 1 year, if he's going to be protected by the vaccine, they have so many years to live and contribute to the development of our nations. So this is the research question: what was genetic factors associated with pediatric HIV disease progression in sub-Saharan Africa? Significance of this is to identify new treatment approaches. All the work that has been done in identifying markers that are associated with delayed progression has led to some development of a new antiretroviral. Of late, I think we have more than 5 classes. Protease inhibitors. We have so many, even inhibitors of fusion and co-receptors. So this work, all those ones are being developed after studies like this. Then also, the vaccine development is important after identifying which genetic markers are responsible for long-term non-progression. Of course, in the short run, what will benefit is, like you have seen, we have in our hospital settings, we have stockouts for antiretroviral therapy. Even when we have a plant in Uganda that is manufacturing ARVs, we keep having these stockouts. So the problem is here. So in the short-term gain can be to prioritize treatment among those in the most need of that. So objective 1, the problem was 4 years ago, we had no well-trained and knowledgeable genomic scientists here in Uganda. So even when 4 years ago I was recruited on this PhD, I was as green as I was then. So I couldn't do analysis on this. So the program had an arrangement to take individuals who had been recruited on this program to undergo didactic genomics and bioinformatics training at Baylor College of Medicine in Houston, and later another university of Cambridge. So these were the seminars that were done during that training program. Courseworks and seminars, many of them are there, ranging from genetics, computational biology, programming, and then bioinformatics, and then ethics for health professions. So this was kind of the training I had to undergo 4 years ago to be able to analyze the work for the subsequent objectives. It also involved a number of laboratory rotations. These were 6 laboratory rotations, each one spanning minimum 4 months. So you'd get attached to a lab and then try to learn as much as what takes place in that lab. And those are the objectives and then the methods of training that we had to undergo then. So results from that kind of work. So following that training, which spanned for 2.5 years, we returned to Makerere here and started answering objective 1, which was, after knowledge acquisition, we had to do knowledge transfer. So we have done, personally, I've done a lot of training through delivering courses here at Makerere. I've also been involved in mentorship programs for bioinformatics, workshops, scientific presentations and seminars, and coordinated some online courses from H3 Bionet, FutureLearn, and Coursera courses. I'm proud to say that over 50% of the students who are here at least have been my students. So if I may talk to Professor Gerald right now, I can say you have a multiplier effect for the investment you have made in me. So secondly, following that training, I've contributed to the development of the graduate bioinformatics training program here at Makerere, and still I can say a number of students here are taking this program here at Makerere. So there's a program Masters in Bioinformatics and PhD in Bioinformatics. I'm happy to say still I contributed to that. And then also through capacity development and collaborative networks, which have been part of the experience I learned from the States. So still from that objective 1, there's a paper that was published in Nature Genetics in Medicine and published in 2017 following the completion of the training. So objective 2, having acquired the experience and the training I acquired from the States, we embarked on data analysis while here. So and the rationale for this objective was to identify the genetic markers that were responsible for pediatric HIV disease progression. And many here we are looking at the common function variants. So the study design here was a little bit complex, but I'll take you through this slowly. So it was a cross-sectional study design nested within a CAFGEN1 project. CAFGEN, like I've introduced CAFGEN in the first slides, is made up of 4 institutions: the Baylor College of Medicine, Children Pediatric Centers. These ones are offering recruitment of these HIV-infected children who we used for our study to collect the data. And then Baylor College of Medicine in Houston offered the genomics training, which, like I've told you, I knew nothing about bioinformatics then and genomics, and I had to go there and acquire this training. And then we had 2 universities, University of Botswana in Gaborone, Botswana, whereas the other part of the PhD students were recruited. And then we have Makerere University, where also a number of students were recruited, of which I was part. And then we had to embark on this journey that I've been describing earlier. So this study would not go ahead without ethical approvals. Ethical approvals were obtained from 3 countries. That's Uganda from these institutions, Botswana from those institutions, and then from the Beda College of Medicine in Easton. And those are the ethical approvals. The study participants have already tried to describe that these were HIV-infected children who are born to HIV-infected mothers. And those who were progressing to AIDS very fast without treatment, we call them rapid progressors. So those ones who are progressing in less than 3 years. And then there are those who are not progressing to AIDS in more than 10 years plus. We call them long-term non-progressors. So just what you can take from this slide, all these children were HIV infected, but not infected through the sexual route, but they are born infected. So what did we do? We did sequencing to look at the genetic mutation that may be responsible for these phenotypes. We looked at the common variation and then the chemokines and tried to analyze per country, then also combined the analysis to find what was interesting there. So the sample size here you can see is what we call the extreme phenotype design, which means you get this cutoff 3 years less, those are rapid progressors, and then 7 years less, and then you pick those at the extreme of this distribution. So if your cutoff is 3 years, you pick those ones who rapidly progress in maybe less than 6 months. That's why you're calling it extreme phenotype design. Then for the long-term non-progressors, the cutoff was 7 years. So we picked those ones who are like maybe the biggest on that, maybe 14 years, 15 years. That's why we are calling it extreme phenotype design. So we had 173 rapid progressors and then 141 long-term non-progressors. So the power was calculated and we detected that if we consider these assumptions, then we can be able to detect about 80%. Our study will have power to detect about 80% differences in those 2 arms. So we used that software to calculate the minimum sample size that we needed to have this study detect the differences at that level. So the main variables for this study were the predictor variables was pediatric HIV infection in these children, and then the outcome of interest was rapid progression of the disease, or what we call rapid progressors, and then the long-term non-progressors. The covariants we considered here were gender and the country of origin, that's Botswana and Uganda. So the laboratory methods that were done for objective 2, we collected blood from the Children's Centers of Excellence, that's Beda here in Kampala, in Mulago, and then Beda in Botswana, performed PCR tests to identify and confirm that these children had HIV, and then did DNA isolation at Makerere University here at the College of Health Sciences and the University of Botswana, quantified and checked the quality of that DNA, then shipped that DNA to Texas, where sequencing was done, and then using at the Human Genome Sequencing Center at Beda, using the instrument called the Illumina 2500 series. So a brief look at the bioinformatics analysis for the sequencing. We converted the B-cell files to the FASTQ files using cassava and demultiplexed the reads also, and then did quality assessment using those tools, then aligned to the human reference version 19, code variants using GATK haplotype column, version 3.5, then did the annotation of those variants using a number of databases such as RIF6 genes, 1,000 genomes, and then GP-SNP and EXAC, and then finally assess the quality of the variants using SNP-F and VCF tools. We also went on to do statistical analysis for this objective using the softwares you are seeing here. EPACS, that does single variant testing and generates some beautiful graphs that you will see. Then also Plink, that did some summary statistics for the variants, and then our statistical computing for fish exactity test and drawing the GG plots, and also did some analysis in locus zoom to visualize the loci where the statistical significant markers were. So from the characteristics of our study participants, we had 150 from Uganda and then 164 from Botswana, and the distribution between the 2 countries for the rapid progressors and long-term non-progressors was not statistically different. So it would be bad if you had a bias in the distribution of the sexes between the 2 groups, among the rapid progressors and then the long-term non-progressors. Because you know, males carry XY and then females carry XX. So we wouldn't want to get biased right from the start. So this was good. We didn't have statistical differences between the two gender in the two groups. Then we also try to see how different were the time to HIV disease progression. So here you can see the minimum or the average time to disease progression among the LTNPs was about 160 months. And then for the rapid progressors was less than 10 months. That was the average time to the onset of HIV-defining disease. So the quality metrics for the exome data, we try to see, okay, our sample has been sequenced, how much coverage has been done. So per sample, we wanted to see, the machine keeps reading the positions. So for LTNP at 20x, how much has been covered per sample? So we had more than 92% were covered. This was good at 20x plus, that is good. And then averagely for the LTNPs also, we have more than about 78% was covered. That was a good coverage for our analysis. And then also the exome kit we are using has been developed in a different population. So we wanted to see how much of the variation we have falls onto this kit. And what you can see for the LTNP, you had about 76% were captured using this kit. And then for the rapid progressors, we had still about 76%. So you don't have any differences in the two sequence data. Otherwise, if you start by having differences here, that means you're also biased. So what do we have here? This is exome sequencing, meaning we are interested in only recording DNA regions of the samples. So 12% of our variants were from the coding regions. That is important. And then we had 482,000, that's about 53%, were missense variants. Missense means they affect the function of the protein. So that was also very important for this kind of analysis. So we wanted to check our variants. Have they been described elsewhere in public databases around the world? And what you see as blue here, per chromosome, it shows you the individual counts of mutations or variants that were missing in any public database. Meaning our samples had novel variation that was worth exploring for identifying mutations that were enriched in these classes. So this is the breakdown of the novel mutations. What you can see is using every database, big database around the world that has human variation, you can see from our samples, we have novel mutations. So our sample was rich in novel mutations. And these are the different variant classes. So still we wanted to see the counts when we distribute or we stratify that per country. So in the combined cohort, we had 399,000 mutations. But for Botswana, we had about 500,000 mutations in the Botswanan population. And then in Uganda, we had about 800,000 mutations of variants. That is common single loci. That means these were common in those populations. They are shared by all the people in that population. Then we wanted to see how many genes have been detected from this data set. In the combined cohort, we had 19,000 genes that were detected. For Botswana, there were 18,000 genes, and then Uganda, 18,900. What you can take from here is we have more variation among Ugandans than the Botswanans. So looking for the common LTNP marker, because this is our point of interest. So we wanted to see which marker was enriched in the long-term non-progressors before we proceed. And what you can see, this graph is called a QQ plot. It shows you the distribution of SNPs in the samples that we are analyzing. But when you see any that is falling off the line is the one that is showing how they are distributed in these samples. Any that falls off shows you it is skewed or enriched in one of the populations. So this was very good. So we wanted to see on what chromosome was this marker. So we used that graph called the Manhattan plot, and it shows us it is on chromosome 11. So that was a very good, interesting finding from our data set. So I wanted to see what population had this variant of interest. So it was a variant on chromosome 11. This was the p-value, nominal p-value for that marker. The odds ratio was 2.2, and it was in 67% of the LTNPs. That was good. And then it was in about 47% in the rapid progressors, and it is in a gene called the MICA, and it is an exonic variant. So we also wanted to say, okay, this is a combined population. How about in the breakdown population? That's for Uganda and then Botswana. So in Uganda, we had the mark that was enriched at prevalence or a percentage of 14.7% among the rapid progressors. And then for Botswana, we had also one enriched at a percentage of 10.7% in the rapid progressors, but totally absent in the Botswana long-term non-progressors. So that was very interesting in that gene. So I wanted to check for these variants. Are they present in public databases? So this shows you, except the one in Botswana, you can see it's extremely rare. In fact, it was almost absent among the public databases. This one tries to show you that people from Botswana hadn't been sequenced to deposit their variation into the public databases. But the MICA, the common variant, was common in the public databases, so it wasn't novel. It had been described before, but not in this status in HIV disease progression. And the one for Uganda, which was also slightly rare because it is at 3% in one of these databases. So somehow that was rare. So what are these common mutations that we found? The one in the combined cohort in the MICA gene is known to cooperate with this pathway called the MAP kinase pathway or ERK pathway, and it plays a role in vesicle trafficking. The immunologists understand this better than I do maybe. But then the one in the Ugandan cohort is found in a region called HLSC on chromosome 6, and it encodes a large number of HLSC alleles, which is responsible in antigen presentation when it comes to infections. And then the one in Botswana, which was exclusively in the rapid progressors, is a protein tyrosine phosphatase enzyme that is known to cooperate also still with the ERK pathway. So what are these pathways I'm talking about? So we have the MICA, the one that was common in the combined cohort. From literature search, we would see that it affects the ERK pathway, and in a way, it leads to reduced activation of the dendritic cells and macrophages. These are antigen-presenting cells, and eventually it leads to reduced viral replication. That's why it is enriched in the long-term non-progressors. How about the PTPN5? It is also known to be associated with the MAP kinase ERK pathway, and still this pathway is important in the antigen-presenting cells. And in this particular population, it ended up being associated with increased viral replication. That was interesting. What we can know from here is dephosphorylation is done by an enzyme called phosphatases, and when dephosphorylation takes place, it leads to increased phosphorylation. There are enzymes called kinases in this pathway. So if the dephosphorylation has been cut down or reduced in the cellular function, then you will have kinases increasing their activity, thereby increasing the viral replication. So, of interest in this common variant, we wanted to see which body cells are known to produce this MICA mutation, MICA protein of interest. And what you can see here, it is mainly the immune cells and also the cells from the reproductive system. Those are the ones from this database. We try to check which cells are being known to produce this protein in high quantities. It's the cells of the immune system and the cells of the reproductive system. So we wanted to see, okay, fine, we have these cells producing this protein. What other proteins do interact with this protein of interest? And what we have, the MICA is interacting with the MAPK, and then these other proteins here. What you can take from here, it is still the phosphorylation and kinases that are playing a role with the interaction of this protein of interest. And then when it comes to PTPN5, it is right here. You can see it is interacting exclusively with the kinases. So this was enriched in the rapid progressors. That means the kinases. And from literature, we found that phosphorylation is one of the processes that is hijacked by the viruses in the cellular machinery to increase its ability to replicate very fast. So this one couldn't be completed without validating or replicating our findings. So we tried to replicate the common variant, the MICA. Unfortunately, it was not able to replicate. These are common things when you are doing genomic studies, but you don't give up. Yeah, so we failed to replicate our mark of interest. So discussion for this objective, the MICA and the MICA mutation, and then the PTPN5 mutations and the mutations in the HLSC region. All of them were predicted as damaging, or probably they are predicted as probably damaging by polyphen 2, and then deleterious by SIFT. So meaning whatever function they are doing, the one that is doing phosphorylation who don't do phosphorylation, ceased to work as a phosphorylating enzyme. And then the one that was doing as a phosphorylating enzymes ceased to work as such. So the marker we have seen is plays an important role in vesicle trafficking, and then also the PTPN5 regulates the activity of the effector molecules in different cells, like we have seen in the previous slide. What we take from here, in HIV disease progression, in pediatric HIV disease progression, there are common variants that were enriched, and they influenced the phenotypes. However, much as we said, we could not replicate that, but at least we are able to say there are common variants that were enriched in the two groups of disease progression. That's the conclusion for that objective. So we have drafted the manuscript that is being reviewed by the PIs, and the title of this manuscript is Common and Rare Variants Important in HIV Disease Progression from Uganda and Botswana Pediatric Populations. So that's a manuscript from that objective, which is under review. So the final objective was to identify new host variants in the chemokines and then the chemokine ligands. So from literature, we know chemokines are being described to be associated with disease progression, HIV disease progression, and they play a very important role. So they had to be addressed here as our third objective. The methods here that were done was whole exome sequencing to identify chemokine variants in rapid progressors and long-term non-progressors, and then we did single loci association tests, as well as the gene burden test to identify those mutations. And what did we do? Like I said, we did single locus association testing and also gene burden testing, and we interrogated all the alpha, beta, gamma, and delta chemokines, and we only found one that had a nominal p-value that was reaching statistical significance using 3 tests. So the test was stringent because we wanted to do it rigorously, so that we don't pick any signal that may not be important. So we took on a map that attained nominal statistical significance for the 2 tests that were done. For the 3 tests that were done. And those mutations were in a chemokine receptor 6. So we wanted to know what is that chemokine receptor 6. So chemokine receptor 6, what you can take from here is a marker for HIV-specific CD4 T cells. So it's an important marker for HIV-specific CD4 T cells. So we also went on to look for presence of the CCR5 delta 32 base pair allele deletion, which has been described all over the world, but it has always been totally absent among Africans. So we tried to run a guess and see, do we have it in our cohort? And we didn't find it. So this agreed with so many studies that have tried to look for it among Africans, and they can't find it. When you have this delta 32 base pair deletion, you will get infected with HIV. That's in case you have 2 copies. But when you have 1 copy, you get HIV, but you can delay to progress to AIDS. So our map of interest in the chemokines was a CCR6, which showed strongest nominal statistical association. It was enriched in the rapid progressors. However, when you try to run another stringent test, which is called collect for multiple testing, it did not pass. So the story ended there. We tried to see, what mutations were there, and we saw out of the 4, 3 of them were neutral mutations in the CCR6. So the conclusion from this is there are non-synonymous SNPs among the chemokines, but they are not associated with pediatric HIV disease progression in Botswana and Uganda. And still we have a paper from that. It's called exome sequencing in 2 sub-Saharan African populations, suggests no role of chemokines and their cognitive ligands in HIV disease progression. Still it is being reviewed by the PIs. So recommendation from my work, HIV disease progression is a complex condition or a complex phenotype. It's not a disease such as sickle cell where you have 1 mutation and you have the disease. A complex phenotype means there are so many genetic factors that may come together to cause the phenotype you are calling longer-term non-progression or rapid progression. So when you're dealing with a complex disease or condition like HIV disease progression, you need you need a cohort that has so many individuals. And here the data I'm presenting is from 314. So our work will progress to analyze a group of about 900 HIV-infected individuals in my next phase. So there is also early this year there's a paper that was published, and it said there's a new, what we call the Pan-African genome that contains 10% extra variation that has not been detected. Our work was using a reference genome that was built mainly some years back, using populations that were not representing much of the African variation. So this is one of the things we will look at when you are analyzing the 900 genomes to see if we can use a reference from the Pan-African genome. Thirdly, there is also, the sequencing is becoming cheaper, and there's a new chip called the H3 Africa Illumina genotyping chip, which can genotype a whole individual genome at $50. That is too cheap. The time we are running our samples here, each was costing $1,000 to sequence one. But now there's a technology of $50 to run one sample. So meaning there's a future for probing. And we saw in my starting slides, we have over 200,000 individuals who are LTNPs in Uganda just alone. So with these two reasons, we can see that probably the next batch of students may be able to take on this challenge at a higher level. So we have students in Uganda and in some 4 institutions in Africa. There was funding from NIH, and genomics and bioinformatics programs have been started. So with combining these two, cheap genotyping assays and then the presence of genomics and bioinformatics programs, I think the next phase is very clear. Based on the time when I started 4 years ago, when there was no one who would do this kind of work. At the moment, we have more than 20 students seated in here who are capable of doing this in the next few years. So the future is clear for these kind of studies to go forward. The limitations of these studies, I had a small sample size, 314 is small for genomic studies that are probing complex diseases such as HIV disease progression. Then also the maternal factors such as how, of course, ART usage, you never know if some of those mothers had started taking ART by the time they deliver these babies. We don't have that information. And in case they are taking ART, where they adhered to the ART, we don't have that information. And then also the children's nutrition status was not captured. Thank you for your attention. I would like to appreciate and give special thanks to a number of collaborators here who I will not mention, but at least all these people have worked with me in this journey, and I'm grateful. Thank you.